Okay, so we are going through the 2019 end of year Sec 1 Express paper. Do take note, I've written some of the answers, so this will actually speed up a little bit. I can spend more time concentrating instead of just focusing on writing. Okay, so first up, let us look at this question over here. It says, figure 1.1 shows some chick cells. So chick cells are actually animal cells. Okay, so these are some of the things you must be thinking through as you are going through the question, viewed under a microscope. So if you look at it, irregular in shape, this is the nucleus. Okay, there are some, you can obviously see the cytoplasm. Okay, and there's obviously the cell membrane over here. Okay, this part over here. Now, of course, the first question, how are these cells different from red blood cells? Do take note, red blood cells are also animal cells. However, they are specialized cells. So what is the first thing that I'm going to highlight in terms of the specialization? So first thing is that these cells actually have nucleus, but red blood cell does not. So really the difference is really about the nucleus for the first item. Next, these cells are irregular in shape, but we know that red blood cells are actually circular biconcave shape. Last one, these cells do not have hemoglobin, but red blood cells, okay, have hemoglobin. So this is some of the differences that you can write. Hemoglobin. Okay, so these are the three differences. You just need to write any one of these because it's only 1 mark. Next, state two similarities between these cells and a plant cell. So plant and cells, animal cells, all of them will have protoplasm, means that they will have cell membrane, nucleus, and cytoplasm. Hence, you just need to give me any one of these two. Out of these three, choose any of the two, and you will get your full 2 marks over here. So quite simple, 2 marks over to get. But key idea, you need to know the difference. Okay, you need to know the difference in terms of animal and plant cells. So knowing the differences, you also know what's the similarity. So when they say these cells, actually the focus is on the idea of animal cells. Those of you who got stuck because they mentioned chick cells and do not know what they are, okay, do take note. If you know that the animal cells, is going to be a lot easier. Next, question 2.2. Question 2. Figure 2.1 and 2.2 shows two plant cells which are placed in different concentration of sugar solution. So think of it, they are placed in a sugar solution, SS sugar solution. Okay, they say figure 2.1, water has moved out of the cell, so water moved out. Figure 2.2, water has moved into the cell. Okay, so now, before we even go into this, now you got to ask yourself, why did the water come out? So most probably, inside will have a higher water potential for the cell, WP for water potential. Outside will have lower water potential. That will be for the case 1. So if case 1 has lower water potential, this must be a concentrated sugar solution. Okay. Now in this case, for this one over here, water is going in. So inside must have a lower water potential. Outside must have a higher water potential. That's why the water is moving in. Okay, so we're going to write down higher water potential. That's why water is going in. Hence, this must be a dilute sugar solution. So, which cell was placed in a concentrated sugar solution? Clear cut, we have the answer. Figure 2.1. Explain what happened in figure 2.2. So, first thing, you must establish which one has a higher water potential. So, the dilute sugar solution, or you can just say the sugar solution, has a higher water potential than the plant cell. That's the first point you must make. Secondly, okay, what is happening? Okay, let me elaborate a bit more. So you must say, instead of saying water moved the cell by osmosis, I think you should write actually the net movement. Okay, that is a net movement of water molecules. Water molecules. Okay, let me erase this. Okay. There's a net movement of water molecules into the cell by osmosis. Because outside higher water potential, then there's a net movement. So after you've written that, next part you need to highlight is that there's actually this idea of water moving from a higher water potential to lower water potential across a partially permeable membrane. Actually, this is the main word here. Okay, so high water potential to lower water potential. That's how the water will move, and it must have this word across a partially permeable membrane because osmosis must have a partially permeable membrane, otherwise it wouldn't work. And hence the cell became turgid. Those who write swollen is actually okay, but turgid is actually the more correct word to use. So take note of all these little details here. Right? This kind of description is actually quite common. Hence, please make sure you know how to go about writing all this movement of substances in terms of osmosis, in terms of diffusion as well. Okay, and even for active transport. These are the three main processes in terms of the movement of substances. Okay, which structure of a plant cell prevent them from bursting when water enters the cells? Answer is actually cell wall. Key idea, because cell wall is inelastic. Because it is inelastic, it actually holds the cell in and prevents it from bursting. Next, question 3. Figure 3.1 shows some plant cells of an onion. So this onion, okay, what you can see here clearly, you can see the cell wall, and the inner one will be the membrane, cell membrane. This one obviously is the nucleus, and of course between the nucleus and the cell wall, cell membrane, this will be the cytoplasm. Actually, these are the only few things you can see. Next, give one evidence to show that these are plant cells. So we know plant cells, they must have cell wall. So the evidence is they have cell wall. So this is the evidence that you will need to identify that they are plant cells. Other than a large central vacuole, okay, these cells are missing one other component of a typical plant cell. So what is unique to a plant cell is, other than a large central vacuole, there's one more thing, okay, that's special to plant cells. Other than the cell wall, other than large central vacuole, what's the last component that's missing? Okay, so it's actually missing the chloroplast. Okay, it's actually missing chloroplast. Now, I thought plant cells need chloroplast to survive, right? Because they need to photosynthesize. But key idea here is, where do you see an onion? So onions are generally grown underground. Okay, sorry for ugly drawing, but onions are generally grown underground. So they're not exposed to sunlight. So key idea here, why it doesn't have chloroplast is really because onions are found underground. Okay, so if onions are found underground, okay, what is the result of it? They will not be able to receive sunlight for photosynthesis. So this is actually quite cool in terms of the idea. Okay, so not all plant cells must look the same. They actually will adapt according to the environment. Next one. Figure 4.1 shows a diver with a bleeding leg wound. Okay, they say the diver is in danger, so obviously the shark is going to eat him, right? So why is he going to be eaten by the shark? I mean, not eaten, but more like chased by the shark. So key idea here, if we want to see the word low concentration, blood cells, diffusion, and high concentration. Okay, so the key idea, the concentration of blood is high at the wound, so concentration is high at the wound, and low in the water away or near the shark. So down here is low concentration at the shark region. Down here will be high concentration of the blood. So that's the first thing you must state. Next, so if there's this difference in concentration, what's going to happen? The blood cells, okay, make sure use of blood cells, since I want blood cells, right? Will actually diffuse from the region of higher concentration to lower concentration. So the blood cells will actually move towards the shark. And hence what's going to happen? Because the shark sends the blood, okay, we actually alert it to swim towards the diver. So this is what's going to happen, and obviously the diver, if he cannot swim fast enough, the shark is going to eat his leg. Next. How is diffusion similar to osmosis? I think in terms of these two, you know that it's about substances moving, okay, substances moving from high concentration to low concentration. So in both process, particles move from high concentration to low concentration. This is actually the first thing. The second thing is they both do not require energy for substances to move. Obviously, by saying this sentence, you know that this is actually with reference to active transport. Okay. Now, why did I say that? It's really because active transport needs to use energy to move the substances. So part 2, how are both diffusion and osmosis different from active transport? I think the first thing I'd like to highlight would be this one over here. So I'm going to copy it from somewhere else. I'm going to paste it here. It's going down too much. Okay, so I'm going to paste it here. So the first idea is this, is that, okay, in both diffusion and osmosis, the particles actually travel from a region of higher concentration to a region of lower concentration. Okay, and this is actually down a concentration gradient. So osmosis and diffusion, you know that it's from high concentration to low concentration. But active transport actually is quite interesting. It's the opposite way. So it goes from a lower concentration. So I'll use a different color to show you green. So for active transport, actually they move from a region of lower concentration to a region of higher concentration. And this is actually against the concentration gradient. So blue is for diffusion osmosis, green is for active transport. So you can see a clear difference here. Of course, the simpler one to write instead of saying this long one is that diffusion and osmosis, okay, do not require energy, but active transport requires energy. What is this energy for? To move the substances. So these are the two differences you could highlight in terms of diffusion and osmosis. Okay? Well, there's only one mark, so you just need to state one of the difference. Next. Figure 5.1 shows some xylem vessels. So xylem, what do they do? So the function of xylem vessels is actually to transport water. Where I think all of us are clear, it transports water. But from where to where? Actually, it transports water from the roots, okay, to the shoots. Okay, the shoots are sort of like the stem and above, okay? So the key idea, you must show me that you know that it's about transporting water. Okay, and next, please highlight this part here. Not just transport any water or how, must tell me from where to where. Now, what is one adaptation that it has and explain how it carries out its function? So there's actually two adaptations here. The first one is that it has no cross wall. No cross walls and protoplasm. Now, what is the purpose of having no cross wall and protoplasm? Okay, this actually helps to ensure smooth flow of water without obstruction. Okay, this is the answer to part B. But there's another adaptation. So we do know that actually the xylems are actually filled with lignin. Okay, what do I mean by filled with lignin? You all remember that the wall of the vessels, okay, have lignin deposited. Lignin actually is like a sort of like a substances that actually help to strengthen the walls. So what does it actually do? Yes, as I mentioned, it helps to strengthen the walls and prevent it from collapsing. So just take note of this. So there are two main functions here. You can list any one of them because they only want one adaptation and its explanation. Last one, xylem vessel is a structure made of dead cells. Why are they known as dead? They are dead because xylems have no protoplasm. Okay, because we know that all living cells must have protoplasm, which means that they have a nucleus. Secondly, they must have cytoplasm. And last one, they should have cell membrane. But in this case here, we do know that the xylem, they have no cross wall, no protoplasm. So therefore, they are actually known as dead cells. So this is a 2019 paper that we've gone through. That's all.